for genotoxicity. The standard battery includes 3 different assays. One is a test for gene mutation in bacteria, which is also known as an Ames assay, a bacterial reverse mutation assay. And as I mentioned earlier, this identifies comparatively subtle effects on chromosomes, typically, point mutations, substitutions, frame shifts, and those types of things. And it employs 5 different tester strains of bacteria that are E. coli and Salmonella typhimurium mutants. There's also an expectation of an in vitro test with cytogenetic evaluation of chromosomal damage, and typically that's done as a chromosomal aberrations assay with mammalian cells, either immortalized rodent cells or human lymphocytes, normal human lymphocytes. And then finally, there's an in vivo test for chromosomal damage using rodent hematopoietic cells. And that's dosing animals in vivo, collecting bone marrow at the end of the exposure period, and then looking for the formation of micronuclei in the bone marrow. And I think I can point one out. So you see this little purple dot in an erythrocyte. That's the incomplete extrusion of the nucleus during the maturation of the erythrocyte from a progenitor cell. And it reflects the occurrence of gross chromosomal damage. Way. So I'll talk next about the safety pharmacology program. Remember, we had the standard battery in genetic toxicity. There's also now a core battery of safety pharmacology assays that are typically expected prior to the enrollment of humans into clinical trials. These are assays which study both in vitro and in vivo effects on the brain, on central nervous system pharmacology, on the lungs or the respiratory system, and on the cardiovascular system. Kurt mentioned in the previous talk some screening assays for hERG binding. I completely agree that these are very useful things to do very early in the course of compound selection and lead optimization, because they give you a sense of either an early kill decision or a way to think about the conduct of the more extensive cardiovascular safety studies that follow both in animals and humans. So this core battery, again, consists of assays in the cardiovascular, respiratory, and neurobehavioral areas. In the cardiovascular space, generally you conduct a GLP hERG assay that's then followed by an in vivo QT assessment. The in vivo cardiovascular safety studies are typically today conducted in conscious telemetrized dogs or monkeys that have probes implanted to evaluate both electrophysiology and hemodynamic parameters following dosing. Respiratory parameters can be evaluated either in these telemetrized dog or monkey studies or in general toxicology studies, or they can be standalone experiments. When they're standalone experiments, they're typically conducted as single-dose studies in a small number of rats. And then neurobehavioral evaluation usually employs either a functional observation battery or an Irwin assay. Again, these are typically done as single-dose studies, most often in rat, and they evaluate a variety of neuropharmacologic endpoints, including muscle grip strength, cranial nerve functions, open field motility, and other parameters. You can have a need to conduct additional safety pharmacology studies based on findings from the general toxicology studies. And some examples of these that are covered in guideline are immunotoxicology studies, renal toxicology, and gastrointestinal pharmacology, such as motility. But the core battery is, again, evaluation of those 3 principal organ systems. So then moving on to general toxicology, the expectations are that new compounds are evaluated in at least 2 species, and at least one of those is not a rodent. Generally, we use the rat as the rodent species, and then use either the cynomolgus monkey or the dog as the non-rodent species, and the choice of which species to use is often driven by some of the data that Kurt was showing, things like in vitro metabolism studies, where you want to make sure that the test species that you're using is exposed to likely human metabolites, and that often will be a guiding factor in the selection of the use of the non-human primate versus the dog, for example. We do have programs where pigs, rhesus monkeys, ferrets, and other species are used as a second species. But most commonly, it's the rat and the dog, followed by the rat and the monkey, and less commonly the mouse and any combination of the non-rodent. So these are normally dosed by the intended route of human exposure. And I think it's important to realize that there's not a single study design or group of studies that fits all molecules. Instead, we tend to tailor the program to, first of all, provide support for the anticipated clinical plan, but we also tailor the approach based on the availability of the active pharmaceutical ingredient, what we know about pharmacokinetics and metabolism between species, the formulatability of the molecule. I noticed we have kind of a gap in our program that we really don't spend a lot of time talking about formulations, but the formulatability of drugs for non-clinical as well as clinical studies is a huge part of drug development. And importantly, one of the attributes of conducting an IND in the US is that the duration of the preclinical studies supporting the human clinical trials is generally in equal units of time. So if I want to do a 3-month human clinical trial, I need to have at least 3 months of coverage in my non-clinical general toxicology studies at the time of opening the IND. Now, if I open the IND and my goal is to do just those human pharmacokinetic studies in the US, I can do that with 2 weeks of dosing, and in some cases with a single day of dosing in both rodents and non-rodents. So the duration of dosing is guided by the indication. It's guided by the anticipated duration of the therapeutic use of the drug. The duration of dosing that we talk about in animal studies, we talk about acute toxicity studies, which are really single-dose experiments in which we're looking for immediate adverse effects on animals, as well as the potential for the emergence of delayed toxicity. So the acute toxicity studies typically have 2 intervals at which we're evaluating safety. An interval that's early, about 40 to 72 hours after dosing, and a follow-up interval about 2 weeks after dosing. Then there's a group of studies that fit into either subacute or subchronic, depending on your choice of terminology. These tend to be 2, 4, or 13 weeks of daily dosing. The sequence of conducting 2, 4, and 13-week studies depends on the requirements for the clinical trial support. So I really like not to do all of those, 2 weeks, 4 weeks, 13 weeks, and there's no regulatory expectation that you do 2 weeks followed by 4 weeks followed by 13 weeks, but you need to have sufficient duration to cover the clinical trial. So most commonly, companies will conduct 4-week studies to give them the greatest amount of non-clinical coverage for clinical trials in phase 1 and phase 2. Those are then followed by chronic toxicology studies, which are by regulation defined as being 6 months in duration in the rodent and 9 months of duration in the non-rodent. I often get asked the question, why do you toxicologists always want to dose so darn high? Because the pressure that toxicology studies put on the manufacturing of API is huge. And the reason is that we're obliged to. The regulatory expectation is that the high dose produces toxicity, unambiguous, clear evidence of toxicity. And the low dose ideally is not associated with toxicity. And typically that's somewhere a modest multiple of what you think the efficacious dose or efficacious exposure is going to be. And the goal there is that you now have the ability to establish an initial therapeutic index. And the middle dose should be something that produces intermediate results that gives you the opportunity to either identify a no effect level or a low effect level. And Kurt described an escalating dose paradigm where you get to a toxic dose and then by definition the next dose below that was the maximum tolerated or the minimally toxic dose. We're really trying to get a sense of the dose dependence of the responses within these studies. I'll just digress for a minute. Some of the most challenging clinical hold issues that I've encountered over my career have been related to instances where toxicology studies had been conducted, but they didn't dose high enough to produce toxicity. And the pushback from a regulatory perspective can be pretty profound. Boy. Okay, so in these general toxicology studies, there are a lot of different endpoints that are being evaluated. We're not just dosing animals and counting bodies at the end of a period of time. The evaluation of toxicity is based on certainly clinical observations. What are we seeing animals do? Those are usually done at least once a day, and ideally at or near the Tmax, the time at which the plasma concentration is at its maximum. We also look at body weight and food consumption. In the shorter duration studies, typically we use daily body weights to look at changes in toxic changes. And as we get into longer duration studies, the frequency of evaluation of body weight or food consumption often is extended out. If I were to have one single parameter that told me, that gave me information, meaningful information about toxicity, it would be body weight. That is one of the most sensitive indicators of adverse findings within these animal studies. We usually do ophthalmic examination in the non-rodent animal, well, in both the rodent and the non-rodent, pre-study and during the last week of dosing. And in the non-rodents, we look at electrocardiography pre-study and during the last week of the study. Continuing on with the endpoints, very key endpoints include clinical pathology parameters. These are typically done, again, pre-study and during the last week in the non-rodent. Because we've got limited blood volume available for evaluation of these parameters in rodents, we usually just look at clinical pathology parameters at or near the end of the study and rely on the comparison of the control animals to the treated animals to see if there are meaningful changes in any of the parameters. The endpoints are hematology endpoints, so total cellular components as well as serum chemistry, clinical chemistry. We look at urinalysis and we look at coagulation endpoints. At the postmortem floor, there are a number of gross observations that we try to glean at the end of the experiment. We do external examinations to include anything that was observed in life. So if there are changes in the skin or bumps that occur or behavioral things that drive evaluation of tissues that otherwise aren't protocol driven, we look at those grossly and collect them. The evaluation grossly is principally that of the external aspects of the animal as well as the body cavities, the thoracic, abdominal, pelvic, and cranial cavities. Once we've collected the tissues at necropsy, often we look at organ weights also as a very potentially sensitive indicator of toxicity. We look at both absolute organ weights and then organ weights relative to the terminal body weight of the animal or the terminal brain weight of the animal. The typical list I've given here, it's a variety of tissues that are weighed at the necropsy floor. If I had my second favorite endpoint that tells me something about general toxicity, it's an organ weight. The thymic weight, particularly in rodents, is extremely sensitive to toxicity, with a significant decrease in thymic weight being evidence of non-specific general stress. Then histopathology. What histopathology is it's the microscopic examination of tissue sections. These are typically formalin fixed, paraffin embedded sections that are stained with hematoxylin and niacin. We try to correlate the microscopic findings with the gross observations. And in the non-rodent species, in the dogs and monkeys, the expectation is that you look at all tissues from all animals. And in the rodent, we typically do things a little bit differently. We've got larger group sizes in rodent studies, and very frequently we start with a comparison of the high dose with the control animals, and then just do a read down of the target tissues in the mid and the low dose animals, as well as the recovery animals. What do we look at? I think if you're past the second row, you can't read this, but the point is that we're looking at a lot of tissues, between 50 and 60 tissues, typically protocol defined in a given study. But that list gets modified based on gross observations and clinical findings during the course of the conduct of a study. Mentioned ICH S3. This is the guidance document that creates an expectation that you look at the toxicokinetics of the drug. The goal here is to verify the exposure to the test material in the dosed groups, as well as a lack of exposure to the test material within the control groups. The TK parameters that are of interest to toxicologists are usually AUC, the area under the plasma concentration time curve, I mentioned here to note the units. Be careful of units. Some kineticists express these as 0 to t, 0 to 24, or 0 to infinity. So you always want to make sure when you're comparing exposure with AUC between studies of either between animals or studies of different duration that the units are the same. Cmax is another critical toxicokinetic parameter. That's the time at which, it's the maximum plasma concentration that's observed. Tmax, the time to achieve the maximum plasma concentration. T1/2, the elimination half-life, and as was mentioned earlier in the day, F is another parameter that we're interested, which is the percent bioavailability. You have to evaluate exposure in animals. That's the bottom line from the ICHS-3. So just a slide on the interpretation of toxicology data. We hear these terms used in project team meetings or in the course of talking about the safety parameters, and I want to just take at least a minute to define what I think of as the meaning of these different terms. So NOEL is an acronym that stands for the No Observable Effect Level. That's the effect at which I can't identify anything that is definitively drug-related in an animal study. No AEL is a modification of that, the No Observable Adverse Effect Level. So often we'll see changes that are drug-related. I might see an increase in a serum transaminase, but it's not associated with injury in a tissue. So it's a drug-related change, but it's non-adverse. Similarly, an organ weight change. I might see a statistically significant increase in liver weight that's 20 to 30% in the increased compared with the control, but it's not otherwise adverse. It's just a bigger liver. LOEL, the lowest observable effect level, another variant on the NOEL. MTD, we had that discussed earlier, the maximum tolerated or the minimally toxic dose. In theory, those are the same thing. Therapeutic exposure multiple, this to me is the exposure at the no observable effect level divided by the exposure at the maximum human dose or the relevant human dose. So the exposure is usually driven by AUC ratios. Again, make sure you've got the same units if you're comparing those. Occasionally, those Cmax, Cmin will define both toxicity and efficacy, and you can express the exposure multiple ratio based on Cmax. There's a nuance with cancer indications specifically, and a guidance document that I haven't mentioned, ICHS-9, has come out over the last couple of years, which describes the terms STD-10 and HNSTD. STD-10 is the severely toxic dose to 10% of rodents, and the HNSTD, the highest non-severely toxic dose in non-rodents. Why is this important in cancer? It's because we expect most cancer chemotherapeutics to have no therapeutic exposure multiple at doses that are going to be efficacious in the clinic. They're going to be toxic in animals. So these HNSTD and STD-10s dose descriptions essentially enable the initial clinical trials to begin at doses that are close enough to the toxic doses defined in animals. So important factors to consider when you're interpreting toxicology data are the ability to identify a no effect level, or a no observable adverse effect level, the potential existence of biomarkers. There will be a talk later today talking about biomarkers for efficacy, and I don't know to what extent we're going to get into biomarkers of safety, but certainly these clinical chemistry parameters, I think everybody's familiar with transaminases as potential markers of muscle or hepatic injury. So if you've got a biomarker in animals that gives you something to look for in people, they can be extremely helpful, and that's one of the major learnings that you're trying to get from the conduct of the non-clinical studies. In most experiments, we're also trying to describe the reversibility of findings. So there are often subsets of animals that go for a post-dosing period to evaluate recovery or evaluate the potential for delayed toxicity. So we want to start defining the potential reversibility of findings. We also have to take into account the likely relevance of a finding to humans. There are some tissues that animals have that people don't have, and there are some tissues that people have that animals don't have. So a direct comparison of the relevance is often can be not achievable. It was mentioned this morning that there were questions about mechanisms of activity. we very seldom know the mechanisms of toxicity. And if we can at least postulate mechanisms of toxicity, those can also be useful in evaluating human risk, because we can start drilling down to potential biochemical pathways or pharmacologic pathways that may be at play in producing toxicity in animals. How much of an exposure multiple do you need? Obviously, there's no one answer to this question, but I feel a lot better when I've got a 100x multiple at my toxic dose compared with my efficacious dose than I do with a 5x multiple. The answer to this question obviously is dependent upon the therapeutic indication. You have a much greater tolerance in certain indications than you do in others. The unmet medical need within the given patient population. The nature of the toxicity observed in animals. I'm going to feel a lot more concerned if I get cardiac necrosis in a general toxicology study than if I get a 10% increase in thyroid gland weight. The ability to monitor that toxicity clinically is very important, and the reversibility, as I mentioned in the previous slide. Importantly, the therapeutic exposure window between the toxic dose and the efficacious dose really never gets wider as you go to longer duration studies. It just gets narrower or stays the same. So from these early short duration studies, even the ones that Kurt would be doing in mice, you want to try to have a therapeutic exposure multiple that's as wide as possible. It gives you opportunity as you extend the duration of dosing to see the emergence of new things. So the IND isn't everything. I don't want to go into all of this, but certainly we had Kurt describe the preliminary evaluation of the safety of compounds. I talked about genetic toxicology and acute toxicity testing with toxicokinetics, safety pharmacology, gene tox screening here, and then our subchronic general toxicology studies. Once you get into human clinical trials, there are other types of toxicology studies that get conducted. Reproductive and developmental toxicology studies, again, these are defined in ICH guidelines. Carcinogenicity studies, these are lifetime studies in rats and mice, again, described in guideline. We're often in a situation where we're trying to define the toxicity of major human metabolites that may not have been represented in the animals. So this is something we spend a lot of time doing, understanding the coverage of the metabolites. Toxicology of degradation products or chemical impurities. As a consultant, I spend probably half of my time anymore looking at the toxicity of impurities. As you switch from one manufacturing process to another, often there are new impurities that are introduced into the API, which were not covered in your initial toxicology studies or your genetic toxicity studies. And this has been I see a nod here in the front, this has been a real bugaboo with regulatory agencies over the past 3 or 4 years with the emergence of new guidelines on the safety evaluation, particularly the genotoxic safety evaluation of impurities. And then toward registration, environmental assessments also. What's the impact of the manufacturing process on potential environmental contamination? These questions get brought up. Where does it go when it's eliminated from the human body, and does it pose any environmental threat? I didn't mention specifically the chronic toxicology testing, but that's typically required before you go into phase 3 clinical trials and studies that enroll a lot of patients for long durations. So I promised a take-home slide. This is the take-home slide. The minimum for the IND, remember the standard battery of gene tox assays, the core battery of safety pharmacology assays, general toxicology studies with toxicokinetics in 2 species that are of duration sufficient to cover the initial clinical trials, and all of these have to be conducted according to GLP. There will be a presentation to follow in the next session that talks a bit more about GLP, so I won't spend any time on it other than to say that it is, GLP is federal law about describing the conduct of toxicology studies to support human clinical dosing. So I'll stop there.
Didactic point. You mentioned red flags as you're going through the studies, like the thymus and the loss of. So one, has to do with what you often see in neuropsychiatric drugs, and that is actually weight gain, not weight loss. So does the standard paradigms used look at, for example, nitrogen balance or whatever, that if you had a weight gain, you'd be able to distinguish what that might be? And secondly, an untoward effect one sees with escalating dose in rodents is seizure. In fact, this is quite common among CNS drugs. So in terms of mechanism of toxicity, do you have any speculations about what may be going on there?
Boy, actually, Kurt, I think you and I have had these conversations over the years. What does weight gain mean, and what does weight loss mean, especially when you're targeting drugs to modify obesity or appetite or other body weight related changes? I honestly have not encountered situations where I've seen an increase in body weight that I would consider adverse. In other words, that it isn't accompanied by other changes. And so my interpretation of an increase is going, you smile, my interpretation in the absence of anything else going on from a tissue standpoint would be that it's non-adverse. It would define an effect level, but in the context of the adverse effect level, it wouldn't. Now, okay, so is that a harbinger for emergent obesity in patients? Who knows? Fortunately, that is a biomarker that we can very readily monitor in clinical settings. There are instances where I have seen increases in body weight that are related to plasma volume expansion with different types of drugs. So it impacts patient compliance because of the weight gain. And are those typically then, is the weight gain typically reflective of the anticipated pharmacologic activity? No. So. Yeah, so there was a question today about distribution within brain. I don't know the answer to whether seizures are harbingers of clinical concern in rodents. And as far as commonly being seen, you know, I work in products for indications that are very, very diverse, and across the spectrum of the types of products, I don't see that very commonly. Seeing seizures in rodents is also something that's kind of hard to do. Requires a very careful evaluation, but it certainly does happen. Do I know a mechanism or what it is? No, I don't. But I would speculate that somehow your drug is distributing within the brain to impact areas that are prone to seizure. I don't have a unifying mechanism.
One of the problems with the weight issue is temporal. When you only do a 13-week study, that may not be long enough time to actually detect something that in humans often takes a lot more. And there are metabolic differences. So in general, I agree, it's been very hard to detect weight gain as a liability. Of that toxicology.
The other problem, just if I can follow up, by the time you take these rapidly growing rats, by the time you get out to 13 weeks and longer, they're if they're being fed ad libitum, they're already extremely obese animals.
There's species issues. But
yeah.
The one question I had was for immunological targets, and in this, as we see this in MS, a lot of times there's a newer use of transgenic models for toxicology because they want to humanize certain aspects of T cells, receptors, subunits. Can you comment on that? How often have you been seeing that? Are there regulatory or other issues with the use of a transgenic rodent species for toxicology?
Most commonly, when we're using the non-clinical evaluation, the anticipated approach in the clinic is to go into human volunteers, so normal humans. And so starting in the non-clinical studies with normal animals is designed to really provide safety evaluation for those normal humans. The other problem that I see with bringing animal models into the normal safety the routine safety evaluation process is they're often not very well characterized in terms of what their spontaneous disease rate is in different organ systems. Often people generate a transgenic because they're interested in a specific signaling molecule in a specific cell type, but they're ignoring what's going on in the testis or in the adrenal gland or other organs that become part of the evaluation. one of the expectations of GLP is that you have a healthy test system during the course of the study. So you don't have dogs that are infected with parasites or monkeys that have tuberculosis, that the test system is healthy. And in many of the transgenics, by design, they're not healthy. And so you have a hard time making a direct comparison. Where we have seen a real application of genetically modified animals in the practice of toxicology over the past 15, almost 20 years has been in using animals that have been made transgenic with human oncogenes or have knockouts of tumor suppressor genes, which then presumably make the animal more labile to tumorigenic effects, and we can shorten these 2-year bioassays down to 6 months. So that's really been the most wide-ranging application of genetically modified animals in the practice of toxicology. Certainly they have application in specific programs, and I've been involved in some of those in the past.
So, Todd, can you tell us how you would evaluate a compound, an orally administered drug that where you have the exposure level peak, even though the dosing you keep dosing it up? Is it as simple as changing the route of administration?
Sorry, so you saturate systemic exposure as the dose continues to rise.
Exactly, yes.
And in that case, you don't achieve enough of a multiple to get into the safety
Well,
valve.
you may not get an MTD.
Right, so the idea is that if I give 10 milligrams per kilogram, 100, or 1,000 milligrams per kilogram, my plasma concentration stays the same, and just more of the compound is going through. So I give the disclaimers, I'm not a chemist, but in my conversations with colleagues in the past about bioavailable compounds, I've heard people say, boy, if you're counting on a formulation change to fix a bioavailability issue, that's a challenge. I certainly have seen examples where you change formulation, you go to spray-dried dispersions or other dispersing approaches that create more surface area for solubilization and therefore absorption. I've seen those have dramatic impacts in animal studies where I might dose a dog at a given dose level with a free base, and then with a spray-dried dispersion at the same dose level, I get 20 times greater exposure. So those things can happen. The reason I started answering your question with the question is, if it happens at significant multiples above where you need to be clinically, then you're actually good, because you can then make an argument to limit your dose, because no matter how much more I put in, I don't get any benefit of increasing exposure.
If you don't see no AL, what happens?
Well, ICHM3, for the first time in a published regulatory guidance, talks about that a little bit. The acceptable reasons for limiting a top dose. One of those can be saturation of systemic exposure or an arbitrary exposure multiple of 50x. So you don't always get an MTD, but you want to be able to have an answer for when your pharm tox reviewer asks you that question in the course of reviewing the IND.
Okay, one last question.
I just want to make a comment using transgenic animals, because especially for Alzheimer's disease it's relevant in the meanwhile. There is request from FDA and also from the European Medical Agency to investigate and to do toxicology in relevant disease models. So for example, if you're addressing A-beta, human A-beta, which is not expressed in a pathological way in a rodent or in a dog, then you have to use studies with transgenic mice so that you can see, for example, target-specific interactions like induction of microbleeds, microedema, or there could be also some toxicity arising by interaction of your drug with your target, and that you can see only in the transgenic. So it's quite an expensive approach, but it's unfortunately requested, and I know because more and more companies are calling us about this request. Also in Parkinson's disease, it's similar. Just a comment.
Yeah, so I don't want to say that that those models are not used. They're not ubiquitously used, and I guess I would come back to the argument that I would make. If you're going to use models which are different from the standard normal animal models, make sure that you characterize the background disease within the models such that you truly can attribute a change to the drug rather than just a spurious observation within the animal species. I've been involved in projects that have combinations of cell-based therapies, gene transfections, small molecule activators of trans genes, all in the same product concept. And of course, you can't do those studies in normal animals because you have to go to either a murine-ized cell-based approach or come up with some way to get past graft versus host versus graft responses. So you do have to tailor the approach to the given indication and the therapeutic strategy.
John, I'll close with bringing it back to neurodegeneration, and I know you're loath to predict a therapeutic index, but for those of us working in this area, doing, let's say, the non-GLP type studies that I mentioned, what would be, in your mind, for a neurodegenerative condition, the minimum therapeutic index that you would want to see to justify continuing with that compound?
Yeah, so again, there's no answer to that other than I would want to see it there be at least a modest multiple.
Define
So,
modest.
3, 5x, something like that, that in these short duration studies, I feel like I've got something that differentiates my toxicity from my pharmacology. I mentioned cancer as a comparison. Often our therapeutic indices in cancer are fractional.
Yeah, but we all know cancer, you can get away with things, but
Yeah.
for an elderly population where it's going to be perhaps dosing for years, certainly months.
Yeah. And my answer is also kind of assuming that the toxicity is related to the pharmacology, with seizures, or it's something that is driven by the compound getting across the blood-brain barrier. If it's if I'm doing that type of a study and I've got myocardial injury or something that is unrelated, then I want the window to be even wider.
Yeah. Okay. Well, thank you, and I think we're ready for lunch.
I look forward to your comments after my talk.